Good morning, everyone, and welcome to the 2023 GRCF Seminar Series. This morning, we are featuring Thermo Fisher Scientific, and the title of our presentation is Digital PCR and QuantStudio Absolute Q Digital PCR System. Our speaker this morning is Marcia Slater. She's Senior Technical Specialist of dPCR and qPCR at Thermo Scientific. And with that, I'm going to turn it over to Marcia.
Well, hi, everyone. I'm Marcia Slater, and as you heard, I'm a technical specialist with real-time PCR and digital PCR. I actually joined through Applied Biosystems almost 27 years ago, and it's nice to be here because one of the very first places I visited as a new specialist at ABI was here at Johns Hopkins and working with the core. So it brings back good memories to be here, and I think this is my first visit back since COVID, so thank you for joining me. So what we're going to go through today is some of the fundamentals of digital PCR. You can advance the slide. So just what is this thing? I like to talk a lot about how digital PCR and real-time PCR compare to each other. I love them both. They both have their place. So understanding how they compare will help you decide for a given project which is the right technology to use. I want to show you the new digital PCR machine, and then throughout, I'll show some of the benefits of using digital PCR in various experiments. Okay, we'll move on.
Move on. Okay.
Yep. And one more. So what is digital PCR? So most people like to think about PCR as having had 3 steps in its evolution. So you could tell just from my time frame that I've probably been doing PCR for a long time. So I started doing PCR back when it was the only option to do bulk PCR. So that meant you were using a traditional thermal cycler and running those products out in an agarose gel. So that was how everything started. And then along in the 90s came real-time quantitative PCR. And so what made that different was 2 things. First, the incorporation of fluorescence. And so we had a way to see the reactions as they were occurring. Then the other development was the machines to actually see that. So the real-time machines, and probably everyone on this call and in the room has used real-time in the past. And then the final step was digital PCR. And while probably many of you are just starting to more recently hear about digital PCR, it's been out for a very, very long time. And so since I said I was kind of having a trip down memory lane here being back, the first time I encountered digital PCR was here. And it was in the 90s. Because it was one of the first authors of digital PCR was the Vogelstein-Kinsler lab. So I remember working with them in their sequencer, and they're like, yeah, we're doing this PCR with all these replicates. And I thought, oh, that's interesting. Little did I know what it would bring me to years later. So let's go ahead and, back up one. I want to stop again. Sorry.
No
problem. I want to give a little more description about this. So with quantitative PCR, I mentioned that we have the fluorescence, and probably all of you are using it. But one of the things you may not have considered with real-time PCR is that you are pretty much always doing a relative quantification. And by that, I don't mean delta delta CT. So the reason why real-time PCR is a relative quantification is that any one of your curves is dependent on comparing it to something else. So if I came to you and said, hey, my sample had a CT value of 28, does that mean anything? So hopefully for those of you who've been doing real time, you're like, well, not by itself. If I came to you and said my treated sample has a CT value of 28 and my control sample has a CT value of 29, could you say something in? The answer is yes. That's a cycle difference. That's a 2-fold change in that gene. So real-time PCR is dependent on comparing to either a reference sample or a standard curve. Even if you're doing absolute quantification by real-time PCR, it has to be against a standard curve, something else. With digital PCR, what you see in that third panel, that represents a single sample. So that's data coming from a single sample. The blue dots are positive technical replicates, and the black ones are negative. So by adding up all of the negatives and positives for a single sample, I can get an absolute quantity. I don't need to compare it to any other sample. So now we're ready to move forward. So It's that breaking the sample out into those individual technical replicates. And there's different names for that. People can call it digitization, microchambers. Sometimes you'll hear the word partitions used, and in some systems you'll hear the word droplets used. But these are all just little individual sub-reactions from that main bulk reaction. So once you divide it out, then the amplification and the fluorescent chemistry are virtually the same as real-time PCR. So it uses all of those same things. But then at the end, you're going to count up the positives and negatives. So in every one of these little sub-reactions, the question you're asking is simply, is it positive or negative? So there's no CT value, just is it there or not there? And then my last bullet point up here, it's actually the second to the last, is how you do that digitization, that creation of all of those technical replicates from the original master mix, the super mix, can have a big impact on your results. One, if it's difficult, you may not get, or you may not enjoy it, but two, you may have variable statistics. You want to have a very uniform breaking out into those individual chambers. The more chambers, the more powerful your statistics. The more uniform each chamber is, the higher the quality of your reaction. As a matter of fact, if they're not uniform, you're not doing good digital PCR. Next slide. So if you've been hearing about digital PCR and you're wondering, is this something that I should look into? So here are some of the times that I've seen that digital PCR can really be a good fit. The first is if you need a really accurate measurement, an absolute measurement of your target, especially if it's something that's a very low level. Digital PCR is a good fit for that. It's going to give you more statistical power than real-time PCR. So if you're looking for very small changes, you may want to jump over to digital PCR. So your real-time machines are probably spec'd for either a 1.5 or a 2-fold discrimination. What if you need something even tighter than that? Digital PCR. If you need absolute quantity, great time to use digital PCR. Down at the bottom, if your samples have inhibitors, that can affect your amplification efficiency. Digital PCR is going to be able to be tolerant of that problem with having potentially inhibitors or other problems that could affect your efficiency. And then my last, where the question mark is, I'm going to hold that till the very end. This is something really kind of cool and different that digital PCR can do. It's a way to answer some complex questions. So I'm going to just give you that little teaser, and hopefully you'll stay with me to the end, because I think it's a really exciting thing that digital PCR can do that qPCR cannot. So let's continue on.
Oops, sorry, give me a moment here.
Okay. So when I think of digital PCR, I break the applications out into two major buckets. What the vast majority of people are using digital PCR for is just an absolute quantification. So they want an accurate and precise measurement of their material. They could be looking at DNA or RNA. And then the areas where this could be used are just about everywhere. So gene therapy is a huge one for digital PCR, but other applications might include copy number variation, viral load. So there are some folks who are quantitating their NGS libraries, because if you don't have a good input, you won't have a good output. Gene editing experiments, and wastewater is a big one. So I mentioned how it's tolerant to inhibitors. So if you can think of a sample type that's probably more prone to have some inhibitors in it, it would be wastewater. So that's why digital PCR is so popular for wastewater. The other main application area I refer to as the rare allele applications. So this is quantifying very small changes, often a SNP or a small indel. It's often a needle in the haystack kind of application. So some of the examples of this, probably the most famous one right now is looking for oncology mutations. So if you do an NGS experiment and you find a driver mutation in that tumor, then the question is, how can I monitor that over time? Doing an NGS run every few weeks is probably not practical, right? So what you can do with digital PCR is identify that mutation and then have a really fast and inexpensive way to monitor that mutation that you found that's of interest. But there's other ways this can be used as well. So it's not limited to DNA. So you could also use it for allele expression. So if you identified some SNPs and you want to see, is there a differential expression between those two alleles where the SNP is? Digital PCR will work. And then the same chemistry would also work for looking at anywhere where you need to quantitate that single base change. I was talking to someone yesterday about microbes. Microbes are not going to be homozygote or heterozygote. They're going to be a percent of one variant or the other. Digital PCR would be the way to go. Next slide. So let's really get into the nitty-gritty of how this really works. So we start out on the left-hand side with a super mixture. So in that tube, you've got your target material, your DNA, your cDNA, or maybe even RNA. You're going to have your TaqMan assay, so that's your primers and probes. You're going to have your digital PCR master mix and maybe a little water. So at this point, you could take that tube and put it onto either a real-time machine or a digital machine. So in real time, the tube would just go straight into your machine. You'll get a CT value or CQ value. With digital PCR, you'll take that material and instead distribute it out into many thousands of little chambers. And your goal, if you can, is to try to get about one molecule of your target into each of those chambers. So that's the specific target of interest. There's going to still be the whole background of total DNA or RNA that's in there. So if you have a single molecule of your target in each of those chambers, there's really no other technology but PCR that you could use to specifically say it's in there, right? There's no microscope that's going to be able to specifically say, oh, that's my gene of interest in that gamish, and there's only one. So in the third panel, we use that PCR and the fluorescent-based chemistries so that after typically 40 cycles, there's enough signal in there for us to say, yes, my target was in that well. So the red ones here are positives, and then the ones that aren't red are candidate negatives. So one of the things that we do with physical wells, which is how we do digital PCR at Thermo Fisher Scientific and Applied Biosystems, is we need to separate empty wells from true negative wells. So an empty well is a false negative. So I would imagine that many of you here have used the dye ROX in your real-time reaction. In real time, it's used for normalization. But in digital PCR, we use this for quality control. We look at the ROX signal in those wells. If the ROX signal is not there, then that means the mixture didn't make it into the well. That's an empty well. It's removed as a false negative. But it's even more stringent than that because we actually evaluate the fluorescent signal of the ROX. We require it to fit into a fairly narrow range. So if it's too low, that might be underfilled, or too high, those are removed as well. It's not worth taking a risk of having a false negative in the reaction. So that gives us confidence that we're getting the real deal. And for those of you who've done digital PCR before, you may already know that the negatives are more important than the positives. So we get that. So we count up the positives, we count up the negatives. Now, for those of you who are new to digital PCR, you're probably saying, but wait a second. Let's go to the next slide. How do I know that there was really only one molecule there? If you're telling me that's going to be the absolute quantity, what if some of those wells, those little chambers, got more than one? And the answer to that is yes, probably a fair number of them did get more than one. So just to kind of show you how digital PCR works, imagine this pitcher is pouring ping pong balls out onto a grid, and they're just going to randomly go. If you let them randomly go into whatever chambers that they're going to wind up with, you're going to get a whole mixture of wells that are either negative, positive, and in some cases have more than one target in there. So we have to account for that in order to have a precise absolute quantity. So the way digital PCR works is it applies the Poisson model to this data. And that's what you're seeing over on that bottom right-hand side with those bars. So it's going to fit the number of positives and negatives to Poisson. Now, the reason why the negatives are so important is that that's anchoring that. That's the only one that we know exactly how many have 0. We don't know exactly how many have 1. We know how many are positive. So then the rest of that is essentially a theoretical fit to Poisson. So that's how digital PCR works. It's using this correction essentially for Poisson to give you the absolute quantity. So that's the foundation of digital PCR. Next slide. So with this system of just counting positives and negatives, there's a lot of goodness that comes out of that as compared to real-time PCR. So I can't see who's online, but I know that there's a fair number of people here that I've trained in real-time PCR. They might have been here for a very long time. But for those of you who I haven't trained on real-time PCR, I'm a real stickler that you should always look at those amplification curves and ask, are they generally parallel? So for those of you on the Zoom, you can't see, I'm doing air quotes, generally parallel. That would tell you that your amplification efficiency is uniform. You can make a good quantitative assessment of that real-time data. So here, what you see in the slide is that these curves are not parallel. Look at the red bars. You can see that they're getting tighter as we go up. That would mean that if I were moving the threshold around, I would be changing my answer because real-time is a relative quantity, right? So down here, the bottom, they're further apart than they are up higher. That's bad news. This is bad real-time data, okay? Because they're not generally parallel. My answer changes if I move the threshold. That's a big red flag. But if this had been two different chambers of digital PCR, this is perfectly fine. And the reason why is at the end, I can see that those two are both positive. So even though they got there through different amplification efficiencies, they still count as positive. So this is perfectly good digital data, very bad real-time data. So that's where when I talked about wastewater, you know, imagine if there was an inhibitor in there that was compromising the efficiency and it wasn't uniform. Digital PCR can overcome that. qPCR cannot. This would be bad. Next slide. Another nice thing about digital PCR, this sounds like maybe kind of silly on the surface, but when you really start thinking about it, it can make your life easier. There's no more CT values or CQ values, whatever you call them. If you've ever had to describe what a CT value is to a collaborator, I see some smiles here. I met, you know, physicians, veterinarians, statisticians, even lay people. You know, you're there trying to say, well, you know, a CT is logarithmic, and the lower your CT, the more stuff. And that collaborator is like, what are you talking about? With digital PCR, your answer is going to be copies per microliter. If you want to be really perfectly technically correct, it's actually molecules per microliter. But you don't need a dictionary for that. If you're working with someone and they don't get what a molecule per microliter is, you know, there's bigger issues, right? So it makes it a lot easier to communicate with the people you're working with. Next slide. I've already mentioned that with digital PCR, we can get an absolute quantity directly from that sample. So if you were doing absolute quantity using absolute standard curves by real-time PCR, then you don't need to do this anymore. You can just go take that one sample, boom, get an absolute quantity. But interestingly, one of the big applications for digital PCR is to quantify standards to do absolute standard curves. So if I was doing a massive screening project with maybe tens of thousands of samples, and I do see that sort of thing, real-time PCR is an easy way to just process those fast and cheap. But the quantity is only as good as the quantification of the standards. So that's why this is a pretty common application, is to see people quantifying their standards, especially for things like a test where you'd have to run a standard. Okay, whoops, wrong way. Whoops. So if you're with me till now, I've already told you I love both technologies. So for me, this slide really kind of brings it all together of how these two technologies fit together in the same world. So what you're seeing here on the vertical axis is your precision, and it's being expressed in a confidence range. So the tighter your confidence numbers are, the better. So you want to be up as high on the vertical axis as you can. That's good. And then across the bottom is the concentration of the target in copies per microliter. We're going from high copy to low copy. So now that we have the axes in mind, the first thing to look at is the dashed lines. That's typical of real-time PCR. And it actually comes much further out over here. It's kind of cut off on the graph. What real-time gives you is a tremendous dynamic range. You can throw just about anything at real-time PCR and get a CQ value. One of the first things I ever did with real-time was I took a plasmid stock. It's like a microgram per microliter. I dipped my tip in, dipped it into my real-time reaction, and got a CT value. It was like 3, but I got a CT value. There was enough material there that I could probably run it straight on an agarose gel. But it's kind of a testament to the dynamic range that you have with real-time PCR. So it can go way out here. And over the bulk of that dynamic range, you have really nice precision. So while it looks a little bit low in this, the way this is graphed, where I am right here, it's really nice. But what if I need more? That's where digital PCR comes in. So digital PCR is this turquoise asymmetric curve. And so there's several key things about this turquoise curve. First is it has some really hard boundaries. So over here at the blue, that's when every single one of the chambers is positive. If every single chamber is positive, I can't figure out the average from that Poisson modeling, so I can't quantitate it. So no precision when I've saturated the system. And then over here, if everything is negative, I can't quantitate that either. There's nothing there. But if I'm able to stay within that dynamic range, I get 2 ways that this can pay off. The first is this peak right here. So this level of precision is something that qPCR just can't do. It can't get to that level of precision. So if you need tighter quantitation than what you were getting with qPCR, you may want to look at that. And that falls when you have an average of about 1 to 1.6 target molecules per chamber. So if you have an average of 1, then about a third of them would be negative. Then the other payoff is right down here. So this downslope, what you see on that downslope is that digital PCR is holding on to higher precision longer than qPCR. So I often get questions about sensitivity. Depending on what you're doing, sensitivity can mean different things. If it's just a matter of detecting something, yes, there or not, I would personally probably go with qPCR. Because if I get 1 copy in my, qPCR reaction, I'm going to see that and be able to say, yeah, it's there. But what if I want to quantify it? What if I need to compare it to something that's also really low? I'm going to have better statistical power out at that right-hand side with digital. So a great example of this was I had a friend at a pharmaceutical company who was doing this massive siRNA knockdown project. So if you came to me and said, hey, I want to do some knockdown work, what would you suggest? You guys probably know the answer, which is, yeah, check your expression changes with qPCR. It's a no-brainer. And that's what he did. The problem that he ran into was that there were a few genes that were so low to start with, because this was a massive project. So they were so low to start with that he didn't have the room to knock them down and have the statistical power to call it. So for any of you who've ever worked and gotten CT values out in those late 20s and into the 30s, you tend to lose your statistical power there. So what he did for those was he flipped just those genes over to digital PCR. So he did all the initial work in qPCR, and then for those that needed it, flipped them over to digital and was able to gain the statistical power to call knockdowns down there. And it was so important that he actually won an award at his company for doing that. So that's a great example of how the 2 technologies fit together. qPCR is great for doing massive screenings, but there's times that you need a little more something. That something can often be met with digital PCR. And then to further illustrate the peak of precision, this is an example. This is data that was run on our past instrument, our 2nd generation instrument called the 3D. And what you're seeing here are 2 assays, and they're each run with digital PCR and qPCR. So hopefully what jumps out at you first off is the gold bars for each assay look the same. Yay, we got the same answer. If we didn't have that, we'd have a big problem. But how these differ between the 2 technologies is on that confidence interval. So if you look at this first one, BK qPCR, that's actually a really nice confidence interval. If that was the type of data I was seeing and maybe I was doing gene expression experiments, I'd be pretty happy with that. but what if it wasn't enough for what I needed to do? What if I needed more? And so that's where you see the digital PCR answer. Again, the answer is the same, but look at that little teeny tiny confidence interval. That's one of the reasons why people will use digital PCR in gene therapy, say to quantitate their viral vectors. They need to have a really accurate and precise measurement of that material. Okay, so any questions so far? I don't know if you have the ability to check the chat.
Nope, we're good.
Okay, so no questions on the chat, no questions in the room. Feel free to stop me anytime if there's questions that you have. I want to switch gears and talk a little bit about quantifying single base changes. So this is the rare allele application. So let's go to the next slide. Okay. So for goodness, it's got to be at least 20 years. Applied Biosystems has had a chemistry for doing SNP genotyping. So in our SNP genotyping chemistry, it's a PCR reaction. You have your forward and reverse primers. And then in order to detect the SNP, there are two different probes. They're labeled with different reporters. So here you see a VIC is the V, a FAM is the F. And then they also differ by that single base change, or it could have been a small indel, something else that you're trying to test for. And during the course of the reaction, these two probes will compete with each other for that common spot. And when the perfect match probe sits down, the enzyme is going to come through and cleave that probe. If the mismatched probe attempts to sit down, it is more than likely going to be pushed aside rather than cleaved. So if we go to the next slide, this has been a very popular and gold standard chemistry for genotyping populations. And next slide. So what you're seeing here, this is really old data. This is from a 7900, and this, I wouldn't be surprised if this slide I have is more than 20 years old at this point. So you're seeing the amount of FAM versus the amount of VIC on these axes. So the dots that we've colored in blue are the samples that had primarily I think that's the FAM one, be cleaved. The ones that are in red had primarily the VIC probe get cleaved. The ones in green had both of them get cleaved. So this is, the green is heterozygotes, and then the blues and the reds are the homozygotes for their respective alleles. And then down at the origin, you see a few no template controls. So this is how this has looked for years. And I would often get questions about, hey, my variant is not a homozygote or heterozygote. It's, a cancer variant, and it might have a certain percent. Could I use this in order to find out how much of it is there? And the problem is that in real time, the answer is no, because real-time PCR was designed to use this chemistry and essentially just bucket out the three groups. It wasn't really meant for you to use that arc and say, oh yeah, there's 40%. And then when you get down to really small amounts, just no way. So this is a great chemistry for genotyping in real time, but it really didn't stand up to do that quantitation that a lot of people started asking for. So next slide. Along comes digital PCR, and it changed the game for this chemistry. So if you think about what would happen, and let me step back a second, grant me a whole lot of artistic license on this slide. It tells what I want to tell you, but I know it's a little bit weird. So imagine on the left-hand side, this is kind of how real time would have looked if you were trying to quantitate a really rare change. So the green ones are wild types, and those rare red ones would be that mutant. And so what would happen if you attempted to do this in real time would be that everything would all cluster together. There's so much more of the wild type that when it tries to plot those two out, it can't even see those reds. They're buried in the noise coming from that excess of wild type. So that was the problem with real time. But over there on the left, I'm sorry, over there on the right, sorry, I'm backwards in the room. On the right, this is what happens with digital. Remember, I said we want to dilute that material out so that we have around 1 molecule of the target or less. And so in the case of these rare allele experiments, the target is both the wild type and the mutant together. So you can see in those chambers of digital PCR, they have germline proportions. So that first one, it only has the wild type. The red one here is by itself. It genotype as if it was a homozygous mutant. So what happens now is because of this dilution of the material, we're able to see those mutants coming out of that cluster that was overwhelming it before. So the way I like to think of this is that what happens in digital PCR is that we're genotyping the chambers. So over in real time, we were genotyping the population. It could have been people, it could have been cattle or fish or whatever your species was. But over on digital PCR, you're genotyping the population of chambers. So that allows you to get extreme sensitivity for these variants. And if we go to the next slide. So you had artistic license on the last slide, but this is actually real data now. So this is how it looks. So let me start over on the left-hand side. This was looking at a KRAS mutation. And so the first panel of KRAS, the Q61H, is where we have a small amount of that mutation, and beside it is its control. So we can see with the first panel, there are 5 little purples, there's 7 of the greens. Those are the ones that had the rare mutation in them. So the purple and the greens are my positives for the mutant. The blacks and the oranges are my negatives for it. And then we do the opposite way. So the greens and the oranges are the positives for the wild type. The purples and the blacks are negative for the wild type. So we get the absolute quantity of both of them. And then that's reflected down here on these graphs. So we're focusing in on just 2 assays up here, but this is looking across a large panel of different rare mutation assays. And what you see here is that we're getting sensitivities that are down at about 0.1%, sometimes even lower, for that rare mutation in these oncology studies. So next slide. So besides the new instrumentation we also have a lot of new assays to facilitate this. So the ones that you just saw, we call our liquid biopsy assays, specifically designed for oncology mutations. And those are guaranteed to get you to at least 0.1% of that rare mutation. So extreme sensitivity, so totally guaranteed for that. So then besides the cancer research assays, there's also assays for doing looking at residual DNA or quantitating viral vectors. But the most important thing I want you to take away from this slide is what's down in pink, the very bottom. Virtually any TaqMan assay can be converted over to digital PCR. So for my friend who was doing that knockdown, he didn't change his assay, he just changed his technology in order to do those. So if you have a well-developed TaqMan assay, I would expect it to convert over to digital as well, with one little asterisk. I'm not going to be able to ask the people on the Zoom meeting, but for here in the room, have any of you ever done primer limiting? So in real-time PCR, primer limiting is a optimization that can be used to improve multiplex real-time PCR reactions. So my asterisk is, don't do that for digital. It's not needed. We actually want to drive the yield, so don't primer limit for digital PCR. Next slide. Okay, so what I also want to share with you is our latest generation machine for doing digital PCR. So I've been doing real-time PCR for more than 20 years. I've been doing digital for more than 10. And so when we first got the news of this new machine coming out, I was at first pretty skeptical, because I know that digital PCR has historically been a complicated, well, maybe not complicated, but tedious workflow. And this machine really changes the game on that. So let's go to the next slide. So when we looked around the state of the art of digital PCR when this machine was in development, we saw a lot of things in both our platforms and other platforms that needed to be addressed and improved. So the first was some of the platforms had a lot of dead volume. So if you were doing that oncology sample, looking for the rare mutation, and maybe you had a fine needle aspirate, there's no sample to waste there. You've got to be able to use as much as you possibly can. Second, we saw that the ability to make the compartments, the chambers, was often very inconsistent. So out in the world, a lot of machines, including ours, claimed that they had 20,000, but in reality, they didn't have 20,000 chambers. When you actually used them, you tended to get less significantly less. I mentioned tedious workflows. That's what I dealt with. What that meant for me was I was there babysitting that experiment the whole day because I had to move it from, the first step, the, generation to the PCR to the reader. If I wasn't there on my time points, that would make my results especially long to get to. But even if I was there, it was a long process. So typically it was 5 hours to get your first data out. And then the last thing is an interesting one. So I've already shared with you that digital PCR is a yes or no at the end. We read the amount of signal. But if you only take one reading, there's some risk. So, with all of the fluorescent machines we have in the lab, fluorescent contaminants can get in there, like dust particles, and alter your results. Well, I shouldn't say for digital to alter. For qPCR, what will happen is it'll elevate your background noise. You'll see it in the multi-component data, but it won't necessarily impact your answer. But in digital PCR, if you only take one shot at asking that question of how much fluorescence is there, you run the risk of false positives if there's a contaminant. So we felt that that was a big one that needed to be addressed. So let's go to the next. So let me start at the right-hand side and come back. So to address that problem of having potential false positives, with this new machine, the plate is going to go in and never move. It's going to go in, stay underneath the reader the entire time. So we take advantage of that and we pre-read that plate. So if there's any type of fluorescent contaminant, dust particles that got on the system or in the system, we read it and that's removed. So, this is really important because with some of the machines, imagine if you saw a reading of 10,000 fluorescent units coming out. You have one data point, and from that you call positive or negative. What if that was a dust particle? If you only have that one read, you don't know. But by having a pre-read, if we see that 10,000 is there, it's like, before it starts cycling, then we can say, aha, that's not due to the PCR. That's something that's contaminating. Remove that chamber from the analysis, not worth the risk. And in addition to the pre-read, it also has algorithms that look at the signal in that well and analyze, does it even look like the real deal? So there's a lot built into this machine to make sure that when you get a positive, it is the real thing. It's extremely fast. So from when you put the plate in and press go, it's going to be about 90 minutes until you get that first data out. The part that blew me away, it is ridiculously easy to use. So I'll show you that in just a moment. It's also extremely consistent. So with past machines, if I got 19,000 chambers acceptably loaded, I would be patting myself on the back. With the second generation machines, that was incredible. With this machine, if you get 19, you actually want to troubleshoot. Something's not right. It is extremely consistent in delivering you virtually all of the wells correctly filled. And then it's also got a very, very low dead volume. So the vast majority of your samples makes it into the chamber to be detected and measured. Let's go to the next slide. Some other key factors about this machine, it's a single machine. So all of the steps of the workflow of creating the chambers, thermal cycling, and imaging are all automated inside of the one instrument. So that's part of what makes it so easy and so fast. It's extremely flexible. So with this machine, I'm going to show you the plate. So this is the MAP 16 plate that we use to run. And you are welcome to, I'll pass it around and take the caps off and on if you want. Be careful with it. It was the very first plate I ever ran, so it's special to me. But your minimum increment run is 4. If you only had a handful of samples today, you could run 1 or 2 columns and then save the remainder unused portion of the plate for later. So that's very budget friendly. So you could run 4 on this plate, as many as 16, working by columns. And then if you need more than that, so forgive me, this slide deck we finalized a little bit before it was announced, but we now have a robotic arm to feed this machine. So if you need extremely high throughput, you can stack plates into the robot and just let it work overnights and over weekends unattended to really maximize your throughput. And then the last key thing on here is that this is a multiplex capable machine. So that's a 5-color machine. We use the rocks for that QC that I mentioned. And then there are 4 channels available to you to multiplex. Now, this is an interesting story, because if there's anyone on the Zoom meeting that I trained in qPCR, I would have been like killing myself to make sure that I discouraged you from multiplexing in real time. So in real-time PCR, truly quantitative multiplexing, it's extremely challenging. You got to do a lot of optimization and validation. And, you know, it takes me back to the very first time I worked with a group that was doing a high-level multiplex. It was a triplex. It was at a pharmaceutical company. It took them a full year with 3 full-time equivalent people to optimize and validate that triplex reaction. So most people don't have a year to kill. Now, that was more than 20 years ago, and they're still using it today. For them, that was a logical investment in time. But for most folks who are not planning to use an assay for years and years, it makes no sense. But the game changes with digital PCR. So with digital PCR, we're trying to get about 1 molecule of target into each of the chambers. That means the competitive effects of PCR are reduced, if not eliminated. So doing a high-level multiplex with digital PCR is actually really straightforward and, for the most part, pretty easy. So I would actually encourage you to multiplex in digital, while on real time, I will be falling all over myself to discourage you from going that route. Unless it's something non-quantitative like pathogen detection.
Did you want the next slide or?
Yeah, you're perfect. Okay, perfect. So I mentioned that one of the things that really blew me away about this machine is the workflow. And I mentioned that I was skeptical. So I've been doing digital PCR for a long time. I knew digital PCR typically required the person in the lab with the magic hands. It required being with it all day. And so when they were showing us this, essentially a slide like this and saying, oh, it's so easy. You just pipette like real-time PCR and put it in the machine and go. It's like, no. There's got to be a catch. Frankly, I couldn't believe it. So I thought, wow, once I ran it myself and couldn't believe it was easier than they led me to believe. I thought, I couldn't believe it, and I work for the company. So if I were in your shoes, I'd be sitting in your chairs going right now like, yeah, I don't think so. So what I did was I worked with a colleague, and go to the next slide. I made a little video of it because, again, to me, this was just mind-blowing how simple it was. And you might need to press again to start my little video. Oops. It's a PDF.
It's a PDF.
Oh, shoot. Yes. That's right. My apologies, folks. This is a PDF of the talk. So what I would invite you to do, you can go to YouTube and type next generation digital PCR workflow, or I'm sure Dave will probably shoot this out because we forgot that we were using a PDF today. My video is 1 minute, 20 seconds, and most of that is just bookend pretty things that Thermo Fisher puts on. It is, I'll describe it for you. It is just so easy. So you'll take that plate that, for those of you in the room, hopefully you've had a chance to look at. You pipette 9 microliters of your super mixture into the port. So that'll be your DNA or RNA master mix and your assay all pre-mixed together. So you pipette 9 microliters of that in, and then you overlay it with 15 microliters of what we call isolation buffer. And then the last step is you put the cap on. That's it. That's it. In previous generations of PCR, I would be taking as much as 5 to 10 minutes per sample. With this, it's seconds per sample. It's better than what all the marketing things tell you. It just blows my mind. So the heart of what makes it so easy is this MAP16 plate. So for those of you who aren't in the room, this is what that MAP16 plate looks like. So what you're seeing here, this is a 16 sample plate. The white squares are where there are 20,480 wells that one sample will be divided into. So we call that an array of wells. So that's why this is an array plate. So each of the squares is where one sample will be divided out into. Underneath those blue caps is where the loading ports are. So those just easily go on and off. And if anybody is counting, there's a extra row of caps just to balance the heated cover on the top. So if we go to the next slide, really, this is the important part of how this works. And I think it's very clever in its design. So what you're seeing in the left, starting out, is the plate, and we're zooming in to where the wells are. The square is a little bit off in the PDF. But in the second panel, the sample is essentially loaded into that big round circle. That's a cylinder on the actual plate for those of you who can't see it in the room. So you load into that cylinder and the sample comes out through these channels. And then into the third panel, you see where the channels connect into the little tiny chambers, the little feeder channel. So with how it's loaded, that aqueous super mixture goes on first, and then the isolation buffer goes on top. The isolation buffer is really just a light oil, so it naturally wants to be on top. And so then we apply air pressure to the system. So if any of you had good eyes, you may have noticed that those caps had holes in them. It's the isolation buffer that's really the caps. So when we apply the air pressure, that aqueous comes out first, and then the isolation buffer comes in behind it. So if you look at these channels in the third panel, it's essentially pushing the aqueous out in front of it, out into those chambers, and then it remains in the channels, isolating them from each other, so there's no crosstalk. So again, I just think that, it's a very clever way to make chambers. And then the reaction occurs and you get positives and negatives. That's what's happening in the right-hand side. So again, 20,480 wells are available for each of the samples. We use a 9 microliter of super mixture as the input. We use a 5x master mix for DNA and a 4x for RNA. So that lets us maximize the amount of sample in that input volume. And then more than 95% of that sample is going to make it into the chambers, because we needed to fight the problems with dead volume that were out there. For some applications, dead volume is not the end of the world, but if you have one of those rare targets in a rare sample, the last thing you want to do is throw your sample in the trash. And that's effectively what dead volume is. And then the fill rate, you should expect to see 20,000 filled for every sample. If I see less than that, I troubleshoot. Next slide. I have a few still images of the software in case you're interested. For those who have done digital PCR, this should hopefully look like an old friend. For those of you who are new to it, what you're seeing here is a representation of the MAX 16 plate in the top left. This was a very simplistic experiment. So what's to the right of the plate is just some simple measurements, the copies per microliter of each of the targets that was in the well, along with its confidence interval. And then the last two columns represent the raw data, which is the number of positives and the total number of correctly filled wells. This was a 4-plex reaction. So you can see that's why there's 4 lines here. We're using 4 dyes. So those were all measured together in the same sample mixture. And then down below, this is the basic view of digital PCR data. These are called one-dimensional plots. They're plotting the amount of signal for each of the dyes versus the well number. And then they color it just to be pretty. The FAM is blue, the VIC is green, Abby, according to their channel, how blue or red they are. So once you've got those laid out, then the dots you see here represent the individual microchambers. The ones that are hovering around 0, those are your negatives. So didn't have much signal happening. Above that, you see a nice cloud of positives. That's a very normal look for digital PCR, because remember, there are some wells that might have 1, 2, 3, maybe more, depending on how much you've loaded in there. So some assays will have a difference in yield depending on your input quantity. So it is very normal to see some spread. I like to think of it as the earth with a cloud above. And hopefully, if everything is going well, you should have a nice gap between that cloud and the ground. And that's where your threshold goes for digital PCR. That separates your negatives from your positives. Everything above is positive, everything below is negative. The computer will set that for you automatically. You can override it, just like you can override your threshold in real-time PCR. Next slide. And then I mentioned that we have these algorithms to check for false positives. If you want, you can toggle that and actually see what wells have been removed from the analysis, which were rejected for having a problem. Usually there's a handful. Next slide. The thermal cycler is integrated into this machine. So you can program the thermal cycler pretty much how you program any thermal cycler. So that's available for you to do. And then your dye choices, your filters are over on the right-hand side. Next slide. And then the last thing about the instrument, it comes with all the normal Thermo Fisher Applied Biosystems service options. So let me pause. Are there any questions so far?
There, any questions in the room? No. Okay, let me check
Check the
here.
chat.
There are no questions on the
Ooh.
chat. You're doing
Hopefully
great.
That means I'm describing everything well.
Yes.
I want you to stay with me because I still have that cool application I want to share with you. So let me give you a few conclusions, and then we'll move on into that cool thing that's kind of, I'm not supposed to share because it hasn't been legally approved by a corporation, but it's so cool I can't help myself. So don't turn me in. So let's go to the next slide.
Oh,
Oops.
whoops, sorry. Give me 1 second.
Okay. So far, what you are hopefully taking away is that digital PCR is a way to get an absolute quantity. You don't need to use standard curves like you do with real-time PCR. You can get extremely high precision if you play within that narrower dynamic range of digital PCR. It has a very low limit of quantification. So that's that low end of the curves that I showed you. And then no CT values when you deal with digital PCR. It's capable of doing rare allele applications where you're looking for that, quantity of potentially a single base change. It's able to get down to at least 0.1% or better of the rare variant. And that's just with a single chamber. You could potentially increase your sensitivity by loading a sample over multiple loading ports. And that can be used for quantifying somatic mutations, doing allele expression, or even quantifying other single base changes elsewhere. Digital PCR is a very robust technology because it's simply an endpoint, yes or no in each of the chambers. So you don't need to have uniform amplification efficiency like you do with real-time PCR. That means it can tolerate inhibitors or potentially difficult sequences that qPCR cannot. Next slide. And then I've also shared with you the system itself. It's a really great system producing high data quality. You get more than 99% of your wells filled with very low dead volume. It's got algorithms and pre-reads to make sure that your positives are truly positives. And then it's capable of high-level multiplexing. Over here in the middle, it's just ridiculously easy to use. I mean, if you try it, you just will be blown away. The throughput, you can go anywhere from as few as 4 to 16 in a single run, and then you can use automation to increase your throughput on a single machine up to about 256 per day per Absolute Q. And our largest configuration is 1 robotic arm with 2 machines. So that takes you over 500 samples per day if you need high throughput. So if you're still with me, let's move on. I want to share with you the thing I'm probably not supposed to be talking about, but it's just too cool.
Am I going? Okay.
So I need to give you a little bit of the history of this for hopefully it to make sense. So when I first started doing digital PCR, most people were trying to do a duplex reaction. And one of the common applications for a duplex was to do copy number variation, where you would have a target gene and a reference gene. And pretty quickly it became obvious that there was a problem doing this as compared to real-time PCR. So imagine you have a molecule that has 3 copies of the segment A on it. And your goal is you want to quantitate how many copies of A's you have in your sample. If you did real-time PCR, you'd get some CT value that reflected that there were 3 A's in that tube. But over on digital PCR, here's the problem. If they're on the same molecule, you could think of those segments as being like tied together with a rope. They're all going to be dragged together into a single microchamber. So even though there were 3 there, my digital PCR counting is off. I only got 1 positive out of that. They were not free to go to different chambers. So with digital PCR, in order to do this correctly, if we go to the next slide, you had to cut them apart. And this is because digital PCR actually counts molecules. So even though we all say copies, 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 it's actually molecules per microliter. So we have to make the molecules and the copies equal to each other if that's what we're trying to test for. So when we cut them apart, CT value is exactly the same. Real-time PCR doesn't care. But look what happened to digital. When they were together, I had 1. Now I've got 3. So you're with me so far on that? Okay, so that's how I thought about molecules per microliter or per chamber for 10 years. And then high-level multiplexing became possible with digital PCR. So let's go to the next slide. So now I can kind of use this in a different way. Because what if I have some piece of DNA that I want to know if it's intact? So a big place where this is becoming popular is gene therapy. Is my construct intact? I've also seen folks who are doing infectious diseases like HIV wanting to know, is that whole genome there or is it fragmented? So I can take that molecule, and next slide, I can design amplicons that span the molecule. And now I can ask, are they all together? Or next slide, do I have fragments? And so on these fragments, none of these fragments has all 4 together. They have subsets. So when it's intact, I should have 4 amplicons being successfully produced in that well. If I have these going in, who knows what I could get? It's going to be random. So next slide. So this is essentially now what I'm looking for in each of those chambers. Am I seeing my chambers that have all 4 and an excess of all 4 compared to what the statistics would dictate? Or am I seeing a subset where, anything's possible in those wells? So here I can see those fragments are just assorting as they want. And then next slide, if you get happy with your PowerPoint, you do something like this. But the point I'm trying to make is, over here, if everything was up to random chance, there is no way my wells would have all 4 in every well, or in almost every well. That didn't happen by chance, right? The fact that all 4 are there means that molecule's intact. Whereas over here, where I'm getting like anything happening, that molecule's fragmented, it's degraded. So this is a great way to not only determine the quantity of that target of interest, but its state. Is it in the configuration that you want it to be? So with this new machine, next slide, we can actually get an export file out with all of those counts that we can use to analyze. So what you're seeing here, so on the header, you see these 25 nanograms, 5 nanograms, and so on. Those are my samples. The first half of this export file is being expressed in copies per microliter. And then we repeat the same samples over on the right-hand side, but now the data is being expressed as the raw counts of each of those conditions. So then if you look at each of the entries, the first row is everything was negative. So I can see over for the 25 nanograms -1, 4,226 wells were all 4 negative. Then we work our way down, single positives, double positives, triple positives, and then the last one I highlighted for you in green, this would be the one where you're asking, are they all there together? Now in this one, they're not. So if I look at these numbers, there's only 284 on the bottom line counts that had all 4. I can see all of the other conditions were substantially higher. So this is an example of a molecule that's not intact, and it wasn't intact. I just did this as an example of how this file looks. But this gives you the tools now to evaluate the condition of your construct or the molecule of interest. So with that, I will leave that there because I just think that is a really exciting thing. And it's something that qPCR can't do. So this is an application that's really unique to digital PCR. The only other way that I could think of to do something similar to this would be NGS. But if I only had a single construct, that's kind of like pulling out the big guns for something where you don't need it. You need a fast answer. Digital PCR can take you there. So we've gone through a lot of things today. I hope you enjoyed the molecule integrity, is what we call that part. Are there any questions?
Are there any questions in the room?
Yes.
So there's just one type of unit right now with and then so for large throughput, that's what the robot's for. There's not any different types of like
Correct.
double thermocycler units, anything. Okay.
Correct. It's a modular system. So what we are seeing is a lot of people who are just starting out just get the basic Absolute Q system. And then if they want to increase their throughput, they can just add the robot. And if they want to really, increase their throughput, they can add a second unit and the robot can feed them both. So one robot can feed as many as 2. So that's where people are going for the extremely high throughput. And then the benefit of that is it's complete walk away automation. So once you have those plates set up, you just put them in, walk away. We've got our stability studies out to, I think we're claiming 72 hours, but if you look at the posters and the data we've done out to about 100 hours. And so far that's for the DNA experiments. For RNA, I'm skeptical of anything with RNA. I'd want to put that in a cold box. So we are doing stability studies for that as well. But if it were me, I would have my robot reach into to a cold box, which is what some people do. At least with real time. Oh, you don't need to go there. That's
Okay.
basic. That if in case you guys, any of you are doing digital PCR, that last slide I had is what I referred to as my holy grail slide. I might as well describe it, I guess, since
Okay, I'll
it
go back
flashed up.
to it. Okay.
When I teach people digital PCR, the most common question I get is, how much do I load on this machine? It's different from real time. With real time, you'll often, load 10 nanograms or something like that. That's not how you want to do digital PCR. So with digital PCR, you want to try to get to the number of target molecules so that you get the best statistical power. And so what this graph is showing you, again, this is the holy grail. So this is the advanced stuff. Hopefully your trainer will use something like this. So the green line represents pretty much the state of the art today, about a 20,000 chamber machine. And then over on the vertical axis, what we're seeing is the confidence. And if you think about a confidence interval, there's a right-hand side and a left-hand side. That's why this is mirror imaged. So it's either side. So made it look pretty to do mirror images. So you want to be as close to that zero line as you can. So with that in mind, if you look at that green line with 20,000 chambers, the closest you get to that zero line is right here with this vertical line. That's at 1.6 target molecules per chamber. So that's like your bullseye on your dartboard. So when I'm training people, I like to train them to shoot for the bullseye. But what I also typically train people is, if you miss this bullseye to the high side, you're pretty close to falling off a cliff. So I will typically teach people that are starting out with digital PCR to actually shoot for right about here. So the bottom is the percent negative wells, and that directly aligns to the average number of copies or molecules in the well. So right about in here is where 1 is. 1 makes for easy math. I like that, right? And that also gives me more room to be higher or lower and not fall off that precision cliff, because the bulk of this really is more of a plateau than a peak. So I like to teach people right about in here. And then these different colors you see represent different numbers of chambers. So as you go to lower numbers of chambers, if you had a poor load, which first off, 10,000 happening on the Absolute Q won't happen. Like it just will not happen. But with previous machines, this was a common thing to see, that you might have lower, you start getting lower and lower and lower. So disregard the red. If you wanted more sensitivity, you could go to more chambers, but at the peak, you don't gain a whole lot in this area. But as you go to low copy number samples, you can see that these are spreading out and you can gain. So that's why I mentioned that if you were doing oncology, adding a second port might boost your sensitivity. But that's a quick run through of my holy grail. If you go deeper into digital PCR, I'm happy to go into more depth, because that is a great tool to be successful with digital. So I will turn it back.
Okay, well,
Thank you.
Thank you, Thermo Fisher and Marcia Slater, for a great presentation this morning. I want to remind everyone that the consumables for digital and qPCR from Thermo Fisher are available at the GRCF core store. And keep an eye out, check our website for upcoming seminars in the 2023 series. Thank you very much, and we look forward to seeing you all soon. Bye-bye.